Hello and welcome back to the final video in this module where we're going to talk about pattern recognition receptors, those innate immune receptors that allow us to detect non-self and typically mount some sort of innate immune response through the expression of cytokines and other effector molecules. So to start talking about pattern recognition receptors in more depth, a first point that I would like to make is that we express many different types of pattern recognition receptors, and we'll sort of learn about various families of PRRs in this lecture today. But before we sort of jump into each family, I'd like to start with the point that we express pattern recognition receptors in distinct locations throughout the cell, which allow us to sample, or you could even say to surveil, various locations for the particular types of pathogens that we are likely to encounter in those sites. So for example, we have particular pattern recognition receptors that are expressed in the plasma membrane and are available to bind to PAMPs, pathogen-associated molecular patterns, that we find in the extracellular environment. And this, for example, would be useful for detecting PAMPs associated with extracellular bacteria and maybe funguses. In contrast, we have other unique pattern recognition receptors that exist in the cytoplasm or the cytosol. And you can notice here that they are frequently interacting with nucleic acid ligands, nucleic acid PAMPs. And this is especially important for the detection of viral infection. And remember that viruses need to replicate their DNA or RNA genomes in the cytoplasm of the cell, which renders them susceptible to being detected by pattern recognition receptors in that location. Finally, TLRs, a particular type of pattern recognition receptor, can also be found in endosomes. Endosomes are another major location where viruses can be found. That's because many viruses actually enter the cell through receptor-mediated endocytosis. That's sort of the way that they find their way into the interior of the cell. So having pattern recognition receptors within endosomes, they're also well placed there to be able to detect viral PAMPs. And so as we'll see, there are particular toll-like receptors that do that function, again, in that particular site. So having said the broad point that there are different pattern recognition receptors for different compartments, let's kind of go through these families of PRRs one by one and look at some of their unique functions and their kind of overall place within this mechanism of innate immunity. We can start with toll-like receptors, or TLRs, and this is probably the most well-described family of pattern recognition receptors. And you can see that there are many of them, and they are sort of restricted or specialized for two major sites in the cell. There are plasma membrane TLRs, which you can see here. These include heterodimers of TLR2 and 6, as well as heterodimers of 2 and 1. Finally, we have homodimers of 5 and homodimers of 4, and each of these are specialized to recognize a particular PAMP that corresponds to a particular type of pathogen that we are likely to see in the extracellular environment. So you can see here, for example, that gram-positive bacteria, they tend to produce something called diacyl lipopeptides, which will activate the TLR2/6 receptor, whereas funguses produce things called triacyl lipopeptides, and that is going to be more complementary to a heterodimer of TLR2 and TLR1. So on and so forth. Flagellin, which is a protein associated with flagellated bacteria, is a strong inducer of TLR5. And flagellin is a really great example of a PAMP because it's something that is very uniquely associated with a pathogen. The flagellum is something that is very classically associated with bacteria. So it makes sense that we would evolve a receptor that could recognize it because it's a very strong non-self signal. Besides these plasma membrane TLRs, we also have endosomal TLRs. And notice that the ligands for the endosomal TLRs tend to be nucleic acid PAMPs, things like double-stranded RNA, or single-stranded RNA associated with viruses, or particular types of DNA known as CpG DNA, which is associated with many types of DNA viruses. The endosomal TLRs are going to help us recognize those various nucleic acid species, especially nucleic acids that we associate with non-self. And one sort of beneficial feature of toll-like receptors being inside of the endosome is that allows them to detect nucleic acids associated with viruses and then not be available for binding to self-nucleic acids, particularly for single-stranded RNA. Single-stranded RNA is in the endosome, most likely it's coming from a virus because our own RNAs are not endocytosed. They don't exist outside the cell. Any RNA that's coming in from outside, and you can think of the endosome really as an extension of the extracellular environment because it's things that we've literally pulled in from outside of the cell. Any RNA that's coming in through that route is likely associated with non-self. And so this allows us to use sort of the physical segregation of those nucleic acid species to further discriminate self versus non-self RNAs. Although we'll see that there are other types of receptors that exist even outside of the endosomes that can detect RNA as well. But for now, we're talking about toll-like receptors. And so the big point here is that the extracellular toll-like receptors tend to be used to recognize large extracellular pathogens like bacteria and funguses, whereas endosomal TLRs are specialized to recognize viruses through the detection of nucleic acid PAMPs. When toll-like receptors are activated, though, that needs to transduce some kind of response. It needs to result in some kind of biological work, an effector function. So we're going to talk about a fairly simplified, but still probably detailed enough for an introductory lesson, the chemical, or I should say the sort of the molecular biological outcomes, the signaling outcomes of toll-like receptor activation. And we might summarize it with a slide like this, where we see that there are various TLRs. And one point to make about TLRs is that they themselves do not have intrinsic capacity to signal. They don't have any intrinsic enzymatic activity. They are not kinases or phosphatases or proteases. They rely, just like cytokine receptors, on downstream effector molecules to sort of transmit or transduce signals into the cell. And pattern recognition receptors, as an overall class, often need the help of adapter molecules to do so. Adapter molecules are molecules that simply serve as physical bridges between receptors and some kind of downstream enzymes. Frequently in the immune system, that's going to be a kinase. The adapter molecules that allow toll-like receptors to associate with downstream kinases are MyD88 and TRIF. And you can see that most TLRs use MyD88 as an adapter molecule. You can see that basically all of the TLRs point to MyD88, with the exception of TLR3, which uniquely uses an adapter called TRIF. And the other exception to the rule here is that the toll-like receptor TLR4, it can uniquely use both MyD88 and TRIF. But for now, what I want to really say is that no matter if you use MyD88 or TRIF, notice that all these arrows end up kind of going to all the same places. There's incredible redundancy and conservation ultimately in the outcome of toll-like receptor signaling. We have so many toll-like receptors so we can recognize so many different types of PAMPs, but really no matter which PAMPs we detect, some fairly stereotyped molecular outcomes are going to occur. And so downstream of these adapter molecules, we're going to get engagement of a couple of major transcription factor families. And again, we're oversimplifying a bit here, but I think, again, I think it's worth just kind of imagining two major transcriptional outcomes of toll-like receptor signaling. That includes the activation of a transcription factor known as NF-κB, nuclear factor kappa B, and it is going to be very important for the induction of inflammatory gene expression. Genes like cytokines and other antimicrobial effectors, which initiate tissue-wide inflammation and have a variety of effector functions that help us control a broad class of pathogens. And remember, this is, in fact, innate immunity. We need a broad-based response. In addition to an NF-κB mediated transcriptional program, toll-like receptors also tend to activate transcription associated with type 1 interferon, and they do that through transcription factors known as interferon regulatory factors, or IRFs. And we saw some examples of these in a previous lecture about interferon signaling, so we'll come back to the activity of IRFs here. So having said that, let's look at these two transcription factors and the programs that they induce, and sort of how do we get from the toll-like receptor to the activation of these transcription factors. Let's first start with NF-κB, which is a very famous kind of canonical innate immune transcription factor. And the way we get to NF-κB activation starts with a toll-like receptor. In this example, there are other pattern recognition receptors that can activate NF-κB, but for now, we're talking about toll-like receptors. And toll-like receptors, through the help of one of their adapter molecules, like MyD88 or TRIF, you can sort of imagine it is about here somewhere in the schematic. But with the help of that adapter molecule, are going to activate a kinase known as IKK. IKK stands for I-kappa-B kinase, meaning that the substrate of IKK is a molecule called I-kappa-B. What is I-kappa-B? I-kappa-B stands for inhibitor of kappa-B. So I-kappa-B, or sometimes just called IKB, serves as an endogenous inhibitor of the transcription factor known as NF-κB, which is itself is a complex. We're just going to refer to it as a single entity here, NF-κB. When I-kappa-B is bound to NF-κB, NF-κB is inactive. It's unable to perform its role as a transcription factor. In fact, it's sequestered in the cytoplasm. It's unable to go into the nucleus. And a transcription factor is not much use in the cytoplasm because it needs access to the DNA in the nucleus in order to transduce any kind of gene expression. However, when we activate a TLR and then we activate IKK, IKK is going to phosphorylate I-kappa-B. That phosphorylation renders it susceptible to ubiquitination, which is going to cause it to be degraded by the proteasome system. So phosphorylated I-kappa-B is degraded, and now this liberates NF-κB, which is no longer bound to its inhibitor. When it is liberated, it is free to translocate into the nucleus, where it can bind to the promoters of genes, and genes whose promoters contain DNA sequences known as kappa-B sites. And so the genes that tend to have kappa-B sites in their promoters are genes associated with inflammation and innate immunity. So when NF-κB goes into the nucleus, it can induce the expression of those immune response genes and thereby create all sorts of innate immune effector outcomes. So this is a general summary of NF-κB activation. And again, this is a major outcome of toll-like receptor activation. However, in addition to NF-κB activation, we also saw that toll-like receptors are strong inducers of type 1 interferon signaling. And so again, a bit of an oversimplified diagram of how that occurs is we have activation of a toll-like receptor, and with the activity or with the help of one of its adapters like MyD88 or TRIF, ultimately that leads to the activation of another kinase known as tank binding kinase 1 or TBK1. One of the major substrates of TBK1 is the transcription factor IRF3, interferon regulatory factor 3. When IRF3 is phosphorylated, it can form functional homodimers, which can then translocate into the nucleus and induce the expression of various molecules associated with type 1 interferon. And you may remember that a major transcriptional target of IRF3 is in fact interferon beta, one of the type 1 interferon cytokines, although there are other interferon-stimulated genes that are controlled by the IRF3 transcription factor as well. So I want to stress that this is not something that's happening instead of NF-κB activation. So this slide and the one before it, these are things that are happening in parallel. We're typically getting simultaneous activation of both NF-κB as well as interferon regulatory factors like IRF3 downstream of TLRs. And this is one of the reasons that pattern recognition receptors can accomplish so many simultaneous pleiotropic innate immune functions, is through a really complex transcriptional program using multiple inflammatory transcriptional factors. Okay, that's about as much as I am going to say about TLRs in this video. And so we can move on down the list to some of the other families of cytokine, or excuse me, of pattern recognition receptors. And we're going to see there's still a lot of convergence in terms of the types of signaling that other types of pattern recognition receptors are going to induce. Another major class of pattern recognition receptors are called C-type lectin receptors, or CLRs. CLRs, because they are lectins, they tend to bind to carbohydrate PAMPs. And this is similar to the lectins that we saw as part of the complement system. There are many pathogens that tend to have unique carbohydrates on their surface that we can use to detect non-self. Yeast and funguses are examples of that, although there are also carbohydrates on the surfaces of bacteria that can activate C-type lectin receptors. I'm giving you one example here of a C-type lectin receptor known as dectin-1, which is good at recognizing beta-glucans, a type of carbohydrate on the surface of funguses. I've really simplified the signaling downstream of dectin-1 here, but just like basically all other immunoreceptors, it also needs the help of a receptor-associated kinase. In this example, it's a kinase known as SYK, which will also lead to the activation of NF-κB, just like we saw previously. So this NF-κB is going to lead to inflammatory transcription. But in addition, simultaneously with the induction of inflammatory transcription through NF-κB, C-type lectin receptors are somewhat unique in that they also induce phagocytosis. And we saw that actually C-type lectin receptors were one of the 4 types of surface receptors that induce phagocytosis on the surface of phagocytes. Here we're seeing them again in the context of immune signaling. So activation of a C-type lectin receptor like dectin-1 is going to induce internalization of a phagosome, and that's also going to be an important effector outcome because it's going to lead to the degradation of whatever we've internalized, like this yeast in this example. So these are C-type lectin receptors or CLRs. Moving down the list, we have another family of pattern recognition receptors known as RLRs. RLRs are found in the cytoplasm, and they are very important for detecting nucleic acid PAMPs, especially those associated with viral infection. RLRs get their name from one of their constituent members known as RIG-I. So RLR stands for RIG-I-like receptor. And I'll point out 2 major RLRs. This is not an exhaustive list, but 2 major RLRs are RIG-I itself as well as MDA5. And notice that both of these molecules have a helicase domain in their structure. Helicases are sort of enzymatic domains which allow interaction with the helix structure of nucleic acids like RNA and DNA. So this is one reason why they are able to interact with, in the case of RIG-I, particular species of single-stranded RNA that have a 5' phosphate group, which sort of is unique to viral RNA. It looks different than the 5' phosphates that we associate with self-RNA. And so RIG-I is very helpful then for detecting single-stranded RNA PAMPs associated with viruses. In contrast, MDA5 is going to be complementary to double-stranded RNA. And double-stranded RNA is a very useful PAMP to detect because if you think about it, when you've learned about RNA in terms of just regular RNA associated with host cells or mammalian cells, you sort of learn that RNA is always single-stranded, and typically it is. We only use RNA for the purposes of translating proteins, and so we never have any reason to have double-stranded RNA. However, that's not the case for viruses. Many viruses are made of RNA, and therefore the only thing that they have to template their genomes to replicate themselves is single-stranded RNA. And then as it is being replicated, there's an intermediate stage of double-stranded RNA as that replication is occurring. So when we detect double-stranded RNA in the cytoplasm of the cell, it's a very clear signal that it probably belongs to a virus and not to the host cell. And MDA5 is a pattern recognition receptor that helps us detect that particular PAMP. In both cases, RIG-I and MDA5 also need an adapter molecule, and the adapter molecule that they share is known as MAVS, or mitochondrial associated antiviral protein. And MAVS is associated, as you can see here, with a mitochondrion, and when these cytosolic PRRs, these RLRs, are activated and associate with MAVS, that leads to the downstream activation of TBK1, which, as we saw before, activates IRF3 and leads to the induction of type 1 interferon-associated transcriptional programs. So you can see here that downstream of the sensors, we have convergent signaling. So there's some redundancy here. We can get to type 1 interferon through multiple means. And again, the reason that we have all of these different pattern recognition receptors is mostly about giving us good coverage for being able to detect many different types of PAMPs. But the signaling outcomes that occur downstream are often very redundant and overlapping because they provide broad coverage across many different types of infections. Okay, so this is RLRs, one type of cytoplasmic pattern recognition receptor. Another major pattern recognition receptor we find in the cytoplasm is a molecule called cGAS. cGAS is a lot more recently discovered and is actually the subject of a lot of ongoing research right now. And I'll give you the highlights of what we currently understand about cGAS. It's a very important cytoplasmic sensor for cytosolic DNA. And this is another example of nucleic acid sort of being in the wrong place and allowing us to sort of know that something is wrong. So for the most part, host DNA in a mammalian cell is going to be sequestered into the nucleus. It's in the nucleus and it doesn't leave. So when we see DNA in the cytoplasm, it's a pretty good indication that it's coming from a virus. Or perhaps it may be our host DNA, but the fact that we're finding it outside of the nucleus is then a sort of danger signal. It's a signal that something is wrong. The cell is probably injured somehow. The nucleus has been perforated or compromised somehow. Therefore, the detection even of self-DNA in the cytoplasm will still activate cGAS and induce an inflammatory response. So whether or not it's pathogen or self-derived cytosolic DNA, you can get activation of cGAS, and cGAS sort of plays by its own rules. Its signaling looks slightly different. Instead of using a receptor-associated kinase, it instead produces a second messenger known as cGAMP, which is a cyclic dinucleotide of A and G. This second messenger, cGAMP, then activates a different signaling molecule known as STING, and STING is found in the endoplasmic reticulum. And when STING is activated by cGAMP, it undergoes a conformational change, which allows it to activate TBK1 once again, which leads to IRF3 phosphorylation, and then therefore leads to the induction of a type 1 interferon transcriptional program, similarly to our other pattern recognition receptors. So you can take note here that the upstream signaling looks a bit different, but again, once again, the ultimate outcomes are very similar with other pattern recognition receptors. And again, I want to highlight that the type 1 interferons, their main function is antiviral effector mechanisms. So it makes sense that no matter how we detect the virus, usually one of the main outcomes is going to be the induction of type 1 interferon signaling. Okay, finally, I want to give you a very simplified, perhaps oversimplified explanation of a final family of pattern recognition receptors known as NOD-like receptors, or NLRs. NLRs are a bit complex, and they themselves also don't really follow some of the traditional rules of immunoreceptors that we've been talking about previous to this. But a major point that I want to make about NOD-like receptors is that they can recognize a broad variety of intracellular danger signals. That includes PAMPs, like bacterial flagellin, as well as cytosolic double-stranded DNA. But NOD-like receptors can also recognize DAMPs, things that are not associated with pathogens, but are signs of cellular damage or just kind of problems within the cell. These include things like indications of pore formation, like ion flux. So if a pore is opened in the plasma membrane, that's going to cause the flux of various cations across the membrane due to osmotic pressures, and a particular NOD-like receptor known as NLRP3 can detect that ion flux. Other things like irritants, like crystals, as well as things like reactive oxygen species, can also activate NLRP3. Whereas more traditional PAMPs are activated, or they tend to activate other NOD-like receptors like NLRC4 and AIM2. A major convergence, though, is that all of these NOD-like receptors, the thing that they have in common is that they form large enzymatic complexes known as inflammasomes. And inflammasomes, from their name, as you can tell, they induce inflammation, and they have enzymatic activity which facilitates that inflammatory process. And the major enzymatic activity that they have is protease activity. So a constituent part of the inflammasome, in addition to the NOD-like receptor, is the protease caspase-1. And caspase-1, we've seen before, one of its main proteolytic targets is a molecule called gasdermin D, which we saw in a previous video was the executioner molecule of a type of programmed cell death known as pyroptosis. So when we have inflammasome activation, that's going to cleave inactive gasdermin D, and leading that activated gasdermin D to form pores in the plasma membrane, which is going to result in a lytic form of cell death known as pyroptosis, which is highly inflammatory. One of the reasons that it is highly inflammatory is that at the same time, the caspase-1 activity of the inflammasome is also going to cleave cytokines that belong to a family known as the IL-1 family. This is a family of highly inflammatory cytokines which typically exist in an inactive form as what we call zymogens. These are molecules that are inactive when they are in their full-length conformation, but when they're cleaved by a protease, they then become activated. The shorter version is activated. And IL-1 family cytokines, when they are cleaved, they are then bioactive where they can then induce inflammation. So the combination of inflammasome-mediated cleavage of IL-1 family cytokines as well as the induction of pyroptosis leads to quite a very strong inflammatory response downstream of the activation of NOD-like receptors. And so these are some of the most inflammatory pattern recognition receptors that can be activated. Okay. So that was the last pattern recognition receptor that I wanted to introduce in this lecture. So let's just summarize the 5 types that we just went through. We started by talking about toll-like receptors, which are found in 2 major locations, the plasma membrane as well as the endosome, and they signal through adapters like TRIF and MyD88, and they initiate some conserved signaling programs through transcription factors like NF-κB and interferon regulatory factors. We saw that C-type lectin receptors are also in the plasma membrane, although they are more specialized for recognizing carbohydrate PAMPs, and they tend to induce things like phagocytosis as well as gene expression. And the induction of phagocytosis is actually something that's unique to CLRs or C-type lectin receptors. Moving into the cytoplasm, we saw that there is a family of PRRs known as RLRs, or RIG-I-like receptors. These are especially important for recognizing viral RNA species, viral RNA PAMPs, and they use the shared adapter molecule MAVs and also tend to strongly induce type 1 interferon signaling. cGAS was a somewhat more unique cytosolic sensor, and it, instead of recognizing RNA, recognizes DNA, and it signals through production of the second messenger cGAMP to activate a molecule called STING in the ER, which then also goes on to produce or to induce the expressions of type 1 interferon. Finally, we saw NOD-like receptors, which are very complicated. I gave you sort of a simplified overview of them. A key feature of them is that in addition to recognizing a broad class of PAMPs, can actually also recognize DAMPs, damage-associated molecular patterns, and other types of environmental triggers. And when they do so, they form inflammasome complexes that accomplish things like the processing of IL-1 family cytokines, as well as the induction of pyroptotic cell death through the cleavage and activation of gasdermin D. Okay, that covers it for Module 3 in the course where we discussed cytokine signaling. This is the last module for this section of the course. From here on out, we're going to shift our attention a bit more into adaptive immunity. I'll see you in the next module.